Hello and welcome to the class on forces changing gene and genotype frequency. We have studied Hardy-Weinberg law of genetic equilibrium. It states that in a large random mating population, in absence of selection, migration and mutation, the gene and genotype frequencies remain constant from generation to generation, and the genotypic frequencies are determined by gene frequencies. Now, if the population undergoes selection, migration, mutation, or is having small size, then the gene and genotype frequency will not remain constant, it will change. Today we will see how selection is going to change gene and genotype frequency. Selection is nothing but differential rate of reproduction. It is choosing of the parents of next generation. The individuals with better performance of a trait are allowed to reproduce, and the individuals with poor performance of the same trait are not allowed to reproduce. In certain cases, particular genotype which is undesirable may not be allowed to breed at all. Before going into the details of selection, we will study one terminology that is known as coefficient of selection, which is represented by letter small s. Coefficient of selection denotes reduction in the gametic contribution of a genotype. We will see three different situations where coefficient of selection is applied against a particular undesirable genotype. In first case, if a particular undesirable genotype is not allowed to breed at all, then S or coefficient of selection is said to be 1 minus 0, that is 1 or 100%. In second situation, if 80% of the individuals of a genotype are allowed to breed, then S or coefficient of selection is said to be 1 minus 0.8, that is 0.2 or 20%. In third situation, if 60% of the individuals of a genotype are allowed to breed, then S or coefficient of selection is said to be 1 minus 0.6, that is 0.4 or 40%. Usually, a breeder applies partial selection coefficient against a particular genotype, as we have seen in second and third situation. Now, we will see one example where we are applying selection against a homozygous recessive genotype a. There are three genotypes at A locus: homozygous dominant, heterozygous and homozygous recessive. All 100% or 1 proportion of homozygous dominant individuals are allowed to reproduce, then the genotype frequency of this genotype after selection will be p square multiplied by 1, which is equal to p square. Similarly, all the 100% or 1 proportion of heterozygous individuals are allowed to reproduce the next generation, then the genotype frequency of heterozygous genotype after selection will be twice pq multiplied by 1, which is equal to twice pq. But only a limited proportion of homozygous recessive individuals are allowed to reproduce, then the frequency of homozygous recessive genotype after selection can be calculated by taking q square multiplied by 1 minus S, which equals to Q square minus Q square S. Here, in this situation, the new total of genotype frequencies after selection can be calculated by adding new genotype frequencies together. That is P square plus 2PQ plus Q square minus Q square S. Here, from this particular equation, it is clear that P square plus 2PQ and Q square is equal to 1 minus SQ square. Now, 1 is the total of genotype frequencies whenever the population is under genetic equilibrium. Because of selection against homozygous recessive genotype, here we can see a proportional loss by SQ square. Now, frequency of recessive gene after selection can be calculated by adding the gametic contribution of homozygous recessive genotype and half of the gametic contribution of heterozygous genotype divided by new total. Gametic contribution of homozygous recessive genotype is Q square multiplied by 1 minus S, and that of heterozygous genotype is twice PQ. Therefore, the frequency of recessive gene small a in the next generation will be Q1 equal to PQ plus Q square multiplied by 1 minus S divided by 1 minus SQ square, which upon solving will yield Q1 equal to Q minus SQ square divided by 1 minus SQ square. Now, the change in gene frequency per generation can be calculated with the formula delta Q is equal to Q1 minus Q. In previous slide, we have seen how to calculate the value of Q1. Q minus SQ square divided by 1 minus SQ square minus Q, the initial gene frequency. Upon solving this equation, we can calculate amount of change in frequency of recessive allele with the formula minus into bracket SPQ square divided by 1 minus SQ square bracket complete. Therefore, the effect of selection on gene frequency is dependent on S, the coefficient of selection, and the initial frequency of recessive allele, that is Q. Based on this talk, there is an exercise for you as your homework. It states that in a population under selection, the initial frequency of recessive gene was 0.2 and the coefficient of selection against the homozygous recessive genotype was 0.3. Based on this information, calculate the change in gene frequency per generation to selection, that is delta Q. Thank you.